The development of molecular biology was one of the greatest achievements in biological science in the 20th century. The discovery of polymerase chain reaction brought enormous benefits, and scientific developments such as genome sequencing, gene expression in recombinant systems, the study of molecular genetics analysis, including the rapid determination of both paternity and the diagnosis of infectious disease. PCR enables the in vitro synthesis of nucleic acids through which a DNA segment can be specifically replicated in a semi-conservative way. It generally exhibits excellent detection limits.
The polymerase chain reaction is a technology in molecular biology used to amplify a single copy or few copies of a piece of DNA across several orders of magnitude, generating thousands to millions of copies of a particular DNA sequence. It is developed in 1983 by Harry Mullins. PCR is now a common and often indispensable technique used in medical and biological research labs for a variety of applications.
The polymerase chain reaction is a method for making many copies of a specific segment of DNA, starting with a very small amount. This technique can be used to identify a specific microorganism from a small amount of DNA, and to identify persons involved in crimes from DNA and cigarettes or in a single hair follicle. The DNA to be amplified is mixed with deoxyribonucleotides, a thermostable DNA polymerase called tag polymerase, and DNA primers. The DNA primers hybridize to the ends of the gene to be amplified and provide the starting point for the tag polymerase. The mixture is heated to break the hydrogen bonds in the DNA, forming single-strand molecules. The mixture is then cooled sufficiently to allow the DNA primers to yield which end of the segments to be copied. Tag polymerase then synthesizes the complementary strand of DNA using the primer as the starting point. The temperature is raised again to separate the DNA strands and then lowered sufficiently to allow the primers to attach. Tag polymerase now synthesizes another set of new complementary strands. This process is repeated until enough DNA has been produced to be identified or used for further research. After 21 cycles, one molecule of DNA can be amplified over 1 million copies. This amount of amplification can be achieved by running the reaction overnight in a thermocycler, an instrument that automatically raises and lowers the temperature at appropriate time intervals.
PCR has been applied to a large number of medical procedures. PCR was for genetic testing, where a sample of DNA is analyzed for the presence of genetic disease mutations. Characterization and detection of infectious disease organisms have been revolutionized by PCR. The spread of disease organisms through population of domestic or wild animals can be monitored by PCR testing. The development of PCR-based genetic fingerprinting protocols has seen widespread application in forensics.
RT-PCR stands for reverse transcription polymerase chain reaction. It is a technique used in genetic studies that allows the detection and quantification of mRNA. It is a very sensitive method that shows whether or not a specific gene is being expressed in a given sample. Reverse transcription is a process of making complementary DNA, cDNA, from a single-stranded RNA. The experiment requires RNA, a reverse transcriptase enzyme, a primer, DNTPase. The RT experiment is also called first-strand cDNA synthesis. Steps are follows. RNA is first incubated with the primer at 70 degrees centigrade to denature RNA secondary structure. This is chilled quickly on ice to let the primer anneal to the RNA. Reverse transcriptase, DNTPase, buffer are added in the reaction. RT reaction is extended at 37 degrees centigrade or higher for 1 hour to allow transcription to occur. RT reaction is heated 70 degrees C to inactivate the enzymes. The end result is cDNA. RT-PCR is a very important test in the field of genetically modified organisms because it gives researchers a mechanism to test whether any specific gene is turned on or turned off. This allows researchers to identify the benefits of genetically modified organisms with respect to their natural counterparts and search for any significant differences in which genes are expressed in the two types of organisms. Nucleotides are arranged in a long sequence, therefore the loop will be formed. It will disturb the process of PCR.